Hi, my name is Andrew. Thanks for putting this symposium on today. First, I want to talk to you a little bit about the problems we've seen with RNA-Seq analysis. We have a website that we put up. It's called RNA-Rocket, and we have more than 1,000 users there. And what we've found is that with the promise of RNA-Seq, so the promise of RNA-Seq is that you can just sequence what's floating around in the cell and see what's going on, as a snapshot, which is a lot more powerful, in theory than microarrays where you have to design what you're looking for. And the problem with the promise of RNA-Seq is people are buying it completely. So they generate their data, or they pay to have data generated, and they think that after they get that data, they should just be able to look at it and see what's going on, and that that's the end of the story. One of the challenges for us is to try to convey to them what they ought to be looking at. So in terms of quality control with respect to their data, a lot of times people will just take their RNA-Seq data, align it to the genome, and they say, oh, okay, now I'm done, now I can see what's happening. And they sort of ignore a common problem, which is the quality of the base calls that they get. So they just sort of, you have this thing in computer science where it's this meme called crap in, crap out, right? So if you start with bad data, you can't get good analysis. And so we've seen tons of users come in and they simply align their genome, and then they don't understand why they're not getting significant results. And so what we try to encourage them to do is to look at the sort of the characteristic tailing in the base calls that come off modern short read aligners, and to look at how much of their so that's the whisker and bar plot on the right, and how much of their sample is actually aligning to the genome. So in this case, red is good and black is bad. So you can see, and here's the base call quality of each of the different categories, or it's the amount of reads mapped for each of the different base call quality categories here. And so if you look, here is a really poor sample. This is from a real user where they didn't understand why they couldn't get good differential expression analysis, and it was because over half their sample wasn't being aligned, and that was because they have really bad base calls. So we try to make users aware of, and here also when you're doing differential expression analysis, if you do have replicate structure, you need to make sure that your replicates are telling you, are in somewhat of agreement, because if they're wildly disagreeing with each other, that's, that kind of doesn't serve the purpose of, well, it serves the purpose to tell you, but unless you look at it, you're kind of missing the point of doing a replicate. So again, we tried to bring the questions that the users face into the interface. We really think that if we just process the data from end to end and just give them the results, we'd be doing them kind of a disservice in terms of making them aware of the challenges they're actually facing. So we sort of bring the questions into the interface, and I just wanted to sort of emphasize that. The other thing I want to talk to you about that we've been working on recently is Pan-genome Graphs. So there's a movement, I think, in both the bacterial space and even the human space, particularly in David Hausler's lab out of UCSC, to regard the genome not just as a flat sequence, a regular string, but as a graph itself, because the human genome even is a composite representation of many individuals. And so by just regarding it as a regular flat string, you sort of miss the variability inherent. So you end up with all kinds of huge VCF files that you have to align against the reference, instead of actually modeling the variation in the reference itself. And so this particular work is for bacteria. And so the idea here is to represent hundreds or thousands of bacterial genomes as a single entity, as a graph in this case. And so this sort of looks like, spaghetti. It looks like a big jumbled up mess. But if I told you, the panel on the left represents chromosome 1 for a composite representation of 400 genomes, and the panel on the right represents the highlighted region, represents chromosome 2, then it starts to make a little bit more sense, and you can actually zoom in. And so we've tried to make this available. I don't have time to go into a lot of detail about how we create the graphs, but we're able to create it in a few minutes and then put it up on the web and stream the information backing thousands of genomes to the user so that they can zoom in and interact and see which regions are highlighted or see which regions are active for any subset of the genomes that you're interested in. So that allows you to do things like rearrangement detection. So in this case, this is, I think, about 60 Brucella suis genomes, and this nexus point highlighted in the middle is the nexus point between chromosome 1 and chromosome 2. And what we found by creating this graph and looking at all the genomes, that almost all the genomes do follow the top two paths that you see here. But then there's one genome which connects chromosome 1 and chromosome 2 in this region of homology between the two molecules. And so what that indicates is that there's probably an assembly mistake. So when you're creating the assembly, you're extending out based on similarity in sequence, and they extended out in the wrong direction, and they created a molecule that didn't exist. So that is one thing that this is able to detect. Another thing is it kind of provides scaffolding for incomplete genomes using the composite information from the other genomes. And the last thing is that it models pretty well inversions, inserts, and deletions so that you can identify rare occurrences of regions of the genome with respect to the group that you're looking at. Okay, that's it for me. Chen Hong's going to speak.